In this module, the basic concepts of crop improvement are presented using trees as model organisms. Provenance trials, also known in the ecological literature as common garden trials, are also introduced. As will be seen, Provenance trial is a powerful tool for the tree breeder and forest ecosystem manager alike, and takes on added relevance in situations where changing environmental/climatic conditions are anticipated. Tree breeding is conceptually simple, but in practice is complex, time-consuming, and expensive. Molecular markers already have utility in tree breeding process for fingerprinting and paternity analysis, applications that can reduce cost and increase gain. The goal of the CTGN project is to further develop the methods and knowledge that will allow genomic tools to be fully integrated into the genetic selection aspects of tree improvement. It is anticipated that such applications would significantly increase selection efficiency and reduce the time it takes to make improvement in artificial populations. The same tools developed for tree breeding should have direct utility for management of natural populations. The long-term goal of a tree breeder is to develop populations that have improved economic traits, are widely adapted, and retain sufficient genetic diversity to deal with future environmental conditions. The tree breeder has a distinctly different perspective to crop improvement than does, say, the corn or soybean breeder, as will be highlighted in coming slides. Genetic gain is achieved by increasing the frequencies of desired alleles in genes that control traits of interest. This ultimately results in reduced genetic diversity. Considering that trees must survive for decades in highly heterogeneous and changing environments, maintaining diversity is quite important. So, the tree breeder must find an appropriate balance between gain and diversity. Historically, it was not always possible to predict how selection for economic traits might influence traits of adaptive significance. Genomic tools may provide means to do just that. Such applications have increased relevance where forest breeders anticipate the use of clones rather than sexually propagated populations in deployed forest plantings. A tree improvement program represents a highly complex array of activities requiring multiplicative skills, physical resources, and long-term planning strategies that can span decades. Before embarking on such an endeavor, it is important to know what your tree improvement and tree breeding objectives are and how you are going to accomplish them. To begin with, it is important to make a distinction between the tree improvement goal and the tree breeding objective. The former is a relatively simple economic rationale for investing in tree breeding, while the latter is a more carefully crafted set of priorities based on both science and economics. The tree breeding strategy employed to meet the tree breeding objective can be very detailed and complex. It is here that the use of markers must be carefully evaluated. Note that the definitions given above are equally valid if the term tree breeding is replaced by land management. A land manager's overall goal is to increase or maintain the adaptability of the plant and animal species on that land, and he/she will define a strategy that will likely include some management process. In the future, that process will undoubtedly include knowledge of the genetics of keystone tree species on the land base. The tree breeder must consider a broad range of issues that ultimately define the improvement strategy. Trees, for the most part, rely on outcrossing with unrelated individuals to produce viable progeny. Selfing, or lesser forms of inbreeding, typically lead to poor survival and fitness. Maintaining pedigrees, something that can be facilitated with markers, is key to long-term program effectiveness. Most traits of interest are controlled by many genes, are influenced by environmental traits, and consequently have rather low heritability. These are all characteristics that lend themselves to marker-assisted selection, or MAS. Tree breeders are fortunate in that most tree species possess abundant genetic variation, so much so that achieving gain can actually be impeded. That is, changing allele frequencies in populations takes more time. Genetic testing in trees may take many years, require large tracts of land, and cost a great deal to evaluate. These are all conditions that lend themselves to mass for improved program efficiency. A first pass at tree improvement often starts with a species or provenance trial. A provenance may be crudely defined as a geographic origin of a group of trees. For instance, the natural range may be divided into provenances which have gross environmental conditions in common. Additionally, stands within provenances and trees within stands can also be identified. Provenance may also sometimes refer to an artificial population created through selection rather than a natural population. For instance, a provenance may be represented by seed collected from 5 to 10 trees in a given drainage, all located within a few miles of one another. In the 1960s and 70s, efforts throughout Europe, Scandinavia, and the UK to evaluate non-native tree species for afforestation began with large species and provenance trials of North American species such as Douglas-fir, lodgepole pine, and Sitka spruce. Seed was collected from throughout the entire range of each species and planted across an array of common garden trials from Spain to Sweden. Provenances were selected for planting at specific locations based on their performance in local trials. Provenance trials represent a great starting point for tree improvement and an excellent means of identifying geographic sources that may be suited to new, anticipated climatic conditions. Such trials can also aid in identifying genes that respond to specific environmental factors. Provenance trials are equally important when planning an improvement program for a native species. As an illustration of diversity in natural populations, note the photo above. Trees are arranged in small blocks representing open pollinated families of lodgepole pine. Color, size, form, survival, and phenology clearly vary dramatically both within and between families and provenances. Could you select 20 trees from this photo and be happy with the diversity you were left with? Traditional tree breeding is shaped by the tree breeding cycle, which typically takes many years to complete. In theory, breeding objectives should stay constant through cycles, but in practice they may change from time to time. To accommodate potential changes, programs need to be flexible and maintain sufficient genetic diversity to change directions. Each of the steps in the tree breeding cycle must be evaluated when developing a tree breeding strategy to ensure harmony and complementation. We will discuss each step in the cycle individually. The base population may represent an entire species or breeding zone or geographic region with millions of trees. The process for selection of trees for the selection population ranges from simple phenotypic appearance in the first cycle to sophisticated techniques driven by breeding values of many traits evaluated simultaneously in advanced populations. Breeding populations may be smaller than the selection population due to availability of flowers for breeding, the application of two-stage selection, or other factors. Finally, a highly select group of trees are identified as those used to produce progeny, or vegetative propagules, for deployment in forest plantations. The selection process determines how much genetic gain can be achieved. It may be simple, such as picking the best-looking tree in a natural stand, or complex, using indices of breeding values and weights, or using multiple stages of selection, as is often the case in advanced generation programs. With rapidly developing genomic tools, we may ultimately be able to supplement traditional methods of selection by selecting directly on genotypes, that is, markers that identify causative mutations. From a quantitative genetic perspective, the goal of selection is to move the mean of the select population relative to the previous foundation population. Consider the illustrations noted here for the trait tree height at rotation age for the seedling and clonal forestry options, that is, additive versus total genetic variation. Given what we learned in the previous module, we can calculate gain from selection as follows. Let's take the seedling case first. We are told that narrow sense heritability, due to additive genetic effects, is 0.2. Selection intensity is approximately 2.0, that is, the best tree in 20 is selected. The population mean is 25 meters, and the population variance is 25 meters squared. We can then calculate gain, delta G, using our gain formula, where gain is equal to the selection intensity times the heritability times the phenotypic standard deviation, or in this case 2.0 times 0.2 times 5, which equals 2.0 meters. For the clonal option, we see that broad sense heritability is twice that of narrow sense, and hence gain due to selection is double that found for the seedling case. The population mean for the subsequent generation is advanced relative to the parent population. It should be made clear here that clonal forestry is not a tree improvement method or process per se, but a tool the breeder uses to capture more genetic gain from the breeding and testing components of the tree improvement process. It is an element of the deployment of gain. This figure illustrates the general concept that selection intensity significantly influences gain expectations, and that selection can occur at several levels, across populations within a cycle and across cycles. It also illustrates that the genetic base or level of diversity continues to decline as you increase selection intensity, as indicated by the narrowing of the triangle as it ascends through the stages of the tree breeding cycle. The base population, or BP, contains all the diversity, but on average has no genetic gain associated with it. That's the first cycle only. Gain increases as the number of individuals in a population decreases in the selection and production populations. With each subsequent generation or cycle of improvement, the base population mean for a given trait is moved, assuming selection is efficient. We close the section on selection by reviewing a topic noted in the previous module, namely indirect selection. The long-lived nature of forest trees requires that most selection occurs early in a tree species rotation, on surrogate traits that predict the actual traits identified in the breeding objective. Desired characteristics of surrogate traits include 1. They possess relatively high heritability. 2. They provide a low-cost, high-precision prediction of the desired trait. 3. They exhibit excellent age-age correlation with the desired trait. And 4. They have no unfavorable genetic correlations with other traits of interest. The use of genetic markers that show clear association with traits of interest generally meet all the categories noted here and must be considered for future tree improvement programs where resources allow. Breeding allows for the remixing of genes, ostensibly leading to fixation of desirable alleles. It is a time-consuming and costly exercise when coupled with testing. There are many breeding strategies and approaches, the choice of which is dictated by the breeder's ultimate breeding objectives. Mating designs determine the parameters that can be estimated. We can classify mating designs by the types of families generated in the process. Incomplete pedigree designs result from open pollinated or half-sib polymix breeding methods. In either of these cases, we do not know which pollen parent sired a progeny, although with markers now available for many species, pollen parentage can be reconstructed. Complete pedigree designs require that we know both parents of a progeny. Conducting controlled crosses is often difficult and costly, but knowing both parents in a cross has obvious relevance given the need to track pedigree over generations. Inbreeding cannot be avoided and must be managed in both the breeding population and the deployment population. There are multiple ways to do this, including judicious use of infusions, subdividing the breeding population, and using mostly unrelated individuals to form the production population. Incomplete pedigree designs may tell us a great deal about the seed parent, but virtually nothing about the male parents. Open pollinated designs, though simple and less expensive, may be biased by certain pollen donors dominating the paternal mix. This can be particularly problematic when seed are collected from different stands where neighborhoods may have significantly different genetic quality. Selfing may be common. The polymix, which is composed of equal amounts of pollen from a known number of male parents, offers the advantage of having a uniform paternal parent array for all seed parents. Potential problems exist if pollen parents contribute or compete unequally. To a tree breeder, the incomplete pedigree mating designs are excellent and low-cost alternatives for conducting backward selection, that is, selecting good female parents based upon the performance of their progeny. Historically, these types of crosses were not good for forward selection, or identifying the next generation of selections, because pedigree of the pollen parent was not known. Molecular markers have changed the equation, obviously. The question then becomes economic. Can one use markers to identify pollen parents posteriorly rather than conducting controlled crosses initially? Mating designs that offer complete pedigree control yield information on both parents and provide for informed control over long-term inbreeding. They are the traditional gold standard of tree improvement breeding programs. The downside to such designs is that they are difficult, that is, costly and time-consuming, to implement. Historically, they have been required to ensure informed forward selection with known pedigree. There are many different complete pedigree designs, each with different strengths and weaknesses. These are just two of many potential complete pedigree mating designs. Brainstorm a little about advantages and disadvantages of these two designs. Consider first which type of design might yield greater information on the genetic potential of a parent. Which would be easier and less expensive to implement? Genetic testing is a core activity of most tree improvement activities, as illustrated in the above figure from White et al., 2007. Field layouts and tests set the limits on the precision of parameter estimates. Tests may serve many functions. 1. Improving propagation populations. 2. Maximizing gain from deployment, that is, matching G by E. 3. Upgrading breeding populations. 4. Optimizing mating designs. 5. Improving the selection index for advanced generation selection. And 6. Evaluating infusions, and so forth. Genetic testing is typically conducted by evaluating progeny created by breeding known parents. Progeny performance is used to identify superior parents for backward selection and to provide plant materials for forward selections. Testing may also be done with clonal propagules and is used to inform selection of superior clones for deployment. There are many types of genetic tests, each designed to address one or more functions and tree improvement objectives. Breeding or mating designs and field testing designs are largely determined by the chosen objective or function of the genetic test. This photo is of a loblolly pine progeny test. A typical field design is a randomized complete block design with each cross planted as a single tree plots, rows, or blocks. The number of locations depends on the anticipated genotype by environment interaction, with more sites required with larger G by E. In a homogeneous region, 2 to 4 sites are typical. There may be 50 or more crosses per test. Individual test sites, therefore, may have as many as 3,000 to 4,000 test trees, plus fillers and borders, and take several acres. It will be managed and maintained for 8 or more years. There are literally millions of trees in such tests throughout the United States. Sometimes, all important objectives can be met with the same breeding scheme and field design. This is true when relationships among selections are minimal. In advanced generation programs with multiple objectives, greater efficiencies can sometimes be obtained by using separate but complementary breeding schemes and field designs for different purposes. For example, replicated field trials might be used to estimate parental performance, while block plots might be used for forward selection. Understanding the genetic architecture of relevant traits is key to optimizing progress. Most of the parameters of interest here were discussed in Module 4. Genotype by environment interaction refers to the differential performance of given pedigrees in different environments, or the genetic by environment interaction. Families that perform well in many environments are considered broadly adapted and can be planted widely. Most families retain a great deal of variation and show relatively little G by E in most species, but not all. G by E may be prevalent in highly heterogeneous environments. Juvenile mature correlations are important if one wishes to make selections at an early age. The faster selections can be made, the greater the genetic gain per unit of time. Genetic correlations are important to ensure that 2 or more traits being selected do not move in antagonistic directions. That is, selecting for progress in one results in a decline in the other. The estimated heritability of a trait is very much dependent on the quality of the genetic test from which it is calculated. A test that does not remove environmental variability well will have lower heritability and poorer resolution for identifying genetically superior individuals. The first and all subsequent generations of tree improvement conclude with an analysis of data collected from genetic tests, leading to the next round of selections. From there, the cycle repeats: breeding, testing, analysis, and selection. Historically, in the first generation of tree improvement, analyses typically were performed using standard de novo approaches. With each passing generation, however, more information on pedigrees becomes known. This information can inform and improve the selection process, and consequently new approaches, such as BLUP, or Best Linear Unbiased Prediction, have been adopted widely. It was not that long ago, until the early 1980s say, that most breeders had to perform ANOVA without the benefit of computers, or later, software that performed mixed models. We would estimate expected mean squares independently. Analytical tools with endless computing power have made the job rather simple by comparison. The genetic gain potential created in the tree improvement TI cycle is ultimately captured and deployed on the forest using propagules generated from the production population. The production population may take one of several forms. In very early stages of tree improvement, seed may be collected from excellent wild stands or seed collection areas, though anticipated gain from these trees is basically nil. Seed orchards, or collections of selected clones that have been vegetatively propagated, typically form the basis of most propagation populations in forest trees. In advanced programs, working with species that can be vegetatively propagated easily, family or clonal forestry is practiced. These approaches require specialized methods and require attention to detail for deployment on the landscape. The seed orchard is the workhorse for forest tree production. The clonal orchard is most common. It is created by selecting individual trees that have proven to produce very good progeny, as demonstrated in genetic tests. Generally, scions from these selections are removed and grafted onto rootstock. The photo shown here is of a grafted seed orchard of loblolly pine that is about 20 years old. The orchard has about a dozen clones, each represented by several copies, or ramettes. Care in the original design of the orchard is critical to orchard efficiency. That is, a highly efficient orchard would have all clones contributing equally to both the pollen and seed pool, with no or little inbreeding or contamination from pollen outside the orchard, a condition that approximates panmixis. The orchard manager strives to maximize seed production, genetic gain, and clonal representation, while minimizing pollen contamination and inbreeding. Occasionally, orchards are established with trees that are still under genetic test. As test results become known, the orchards are rogued or reduced in number of clones, a point at which markers may play a role in the selection process. Selections obtained from orchard clones are wind-pollinated, typically, and may be deployed as a mixed seed lot or as families. Seed from a single clone would represent a half-sib family, where only one parent is known. In the U.S., Full-sib family forestry has been practiced to a modest degree in pine and Douglas-fir, often in combination with rooted cutting programs. This is typically done using systems such as controlled mass pollination or supplemental mass pollination. Seed from such crosses, made in the seed or breed orchard, are subsequently sown and used to create multiple copies of the same genotype through rooted cuttings. The photos above illustrate seedlings obtained from controlled crosses that have been grown briefly and sheared or hedged to produce many shoot tips. For a few years, these shoot tips retain juvenile characteristics that favor rooting of cuttings. Serially hedged mother stock plants can provide large numbers of plantlets in a few years. The diploid population is a conglomerate of clones derived from a relatively few crosses. However, the cost per tree planted is relatively high compared to seedlings. Gain from full-sib family forestry with multiplication must be significantly greater than with seedlings alone to offset the upfront cost of vegetative multiplication. Deployment of half or full-sib families provides greater flexibility in matching genetics to environment, if genotype by environment interaction is a concern. The most aggressive form of forestry relies on the planting of clones representing the very best selections. Relatively few species are amenable to large-scale vegetative reproduction, but there are remarkable examples of clonal forestry around the world, including eucalyptus, acacia, and poplar. There are currently some 60,000 acres of clonal poplar in the western U.S., including the trees shown here that are growing on lands once considered desert, but now irrigated with water from the Columbia River. Clones are typically planted in pure blocks, as is obvious in this photo. Typically, a single land manager will plant as many as 10 or more clones to ensure some level of genetic diversity is retained. Cloning allows for the maximum capture of genetic gain, that is both additive and dominant genetic variation, and maximum flexibility in matching genotype to environment, but it results in the least genetic diversity deployed on the land. From this comparison, it is pretty clear that an increase in use of deployment options that favor greater gain will result in a rapid decline in the amount of genetic diversity retained in the forest. How much incremental gain can be achieved using family or clonal forestry? It depends, but doubling the gain of seedling deployment would not be unusual. How much diversity is lost in doing so? Again, it depends on many things, but even 20 clones would retain the vast majority of variation found in a typical forest tree species. What are typically lost in such cases are rare alleles. Much of this diversity is still available in the breeding program and can be used in subsequent deployment populations should its use be warranted. Formal tree improvement programs around the world have relied on the cycle of breeding, testing, selection, and production for the better part of the last 70 years and have made remarkable gains for most traits targeted for improvement. However, the gains made have occurred at considerable cost and time invested, and in recent years, support for large integrated tree improvement programs has waned. Many private companies have abandoned or significantly reduced their tree improvement efforts, and large tree improvement cooperatives have been forced to find ways to do more with less, or find better ways to do traditional activities. Consider a single large tree improvement program in British Columbia, responsible for the domestication of more than 10 conifer species. The challenges of conducting and maintaining the genetic testing element of this program are enormous. Clearly, finding ways to do things better and cheaper are needed for forest tree improvement. Genomics provides many potential applications for the modern tree breeder. We close this module with a glimpse of where in the tree improvement cycle marker-assisted selection may play a role in improving selection efficiency, increasing gain, or reducing the time and cost of conducting tree improvement activities. These points are indicated by the red lettering that refers to marker-informed breeding or marker-informed breeding opportunities. We will discuss this slide at much greater length in future modules.